I'm going to share my screen. Okay, so for question number 4, I hope you have done the question. Okay, so question number 4. Alright, so if you receive this sort of question, okay, where you have a paragraph of question and there's a lot of information to digest, so you read the question and then you extract out all the information given and write it in a corner. Okay, so basically in this question it tells you that the inheritance of cat breed, which is Munchkin. Okay, so the information given in this question, it says that the dominant M allele, okay, will it will express cat with very short legs. Okay, and the recessive M allele will express the normal legs, okay, for the cat. But if the cat if the fertilization between egg or sperm, okay, that carries both of the dominant allele and produces zygote, that is in the condition of homozygous dominant, so the zygote will not be able to survive in the womb. Maksudnya matilah. Okay. And for the homozygous recessive, this produces normal kitten. And for the heterozygous, it produces kitten with short legs. So all this information are given in the question. So you just extract out all the information right in the corner, so that when you do the crossing, when you do the Punnett square, it is easier for you to determine the phenotype after you have the genotype written in the Punnett square. Okay. So if you look at the last sentence, it says here, shows the ratio of viable normal leg kittens shall be born if their parents are both heterozygous for the Munchkin gene. So both parents are heterozygous. So heterozygous is capital M small m. So you just fill up the diagram given in your workbook. Okay, so heterozygous, capital M small m for the female parent, capital M small m for the male parent, and then you separate the allele into separate gametes. Okay, so you have, and then just fill up the Punnett square lah. Okay, and then just combine the fertilization between eggs and also sperm. So you will get capital M homozygous dominant, which will not be able to survive. And then you have two heterozygous, so it means that 2 kitten will have short legs and 1 kitten will have normal legs. Okay, so the question asks for you to show the ratio of viable normal legs. So 1 normal to 2 short legs. Okay, so kita tak kira yang mati ni. So selalunya pecahan tu di per 4 kan, per 4, 2 per 4. Okay, so for this type of question, it already tells you that for homozygous dominant, the kitten will not be able to survive. So only 3 offspring produced. Okay, so 3 lah. So 1 normal legs ataupun 2 short legs. Ataupun you can also do it in ratio. Sorry, in percentage, which is 33%. Okay, 33%, which is 1 out of 3 will have normal legs. Okay, normal legs. So when you are writing ratio, jangan tulis nanti 1 nisbah 2. 1 apa? Nisbah 2 apa? So you must write 1 normal to 2 short legs. Mesti disertai nombor tu, ratio tu mesti disertai dengan dia punya phenotype. Okay. So boleh faham Okay. Alright, so for question B, figure 13 shows DNA replication. Okay, so identify strand A and strand B. So this is easy. So strand A will be leading strand. Okay, strand B will be lagging strand. This is easy. you just have to identify the type of strand. But addition to you identifying the strand, you have to know the feature of this strand. For the leading strand, what are the feature? It is synthesized from origin of replication towards replication fork. Okay, so for the lagging strand, it is in the opposite direction, where it is synthesized from the replication fork towards origin of replication. Or you can also write it as it is the lagging strand or the Okazaki strand, it is synthesized away from replication fork. That is how you describe the synthesis of the lagging strand. And another feature of leading strand, you have to know all these features. The question may ask you the features. Okay, so for leading strand, it is synthesized continuously. Okay, continuously in the 5 to 3 prime direction, and it only requires one primer. Okay, it only requires one primer. And as for the lagging strand, it is synthesized discontinuously as Okazaki fragments. Okay, it is synthesized discontinuously as Okazaki fragment. And it requires many primer. Okay, it requires many primers as the synthesis of one Okazaki fragments requires a primer. Okay, so you have many Okazaki fragments, so the synthesis of lagging strand will require many primers. So you have to know the difference between these two strands. Okay, the another question may ask you to differentiate. Okay, to differentiate maybe the leading or lagging strand. So you have to know the features of these two strands. Okay, so so this usually for the first question and the first few questions, it is really it is easy. You just have to identify or give definition, for example. Okay, so that is straight from your notes. Okay, so this is what I so the one that I've just explained is for question number 2 actually. Okay, so question number 2, state the difference between strand A, strand B. Okay, strand A, you know that it is leading strand. Strand B, it is lagging strand. So strand A, as I said before, it is leading strand. It requires only one primer. Okay, for strand B, it requires multiple primers. Okay, multiple primers. Okay, one Okazaki fragment will require the synthesis of one Okazaki fragment requires a primer. So you have many Okazaki fragments, so there will be many primers. Okay, so for the leading strand, it is synthesized continuously. Okay, from origin of replication towards replication fork. Or in another way of explaining it is that the leading strand, it is synthesized away. Okay, away from origin of replication or towards replication forks. Mana-mana boleh, asalkan awak faham. Okay, tak perlu hafal pun, asalkan awak tengok gambar ni, awak dah faham dah macam mana nak explain. Okay, and then for the lagging strand, it is synthesized discontinuously as Okazaki fragments, and the synthesis is away from replication fork, or another way of explaining it, it is towards origin of replication. Okay, so from this diagram, okay, you have to know that for the synthesis of Okazaki fragment, they have to be joined together to form a continuous lagging strand. So what is the enzyme requires to join the Okazaki fragments to form a continuous lagging strand? The enzyme required for you to join the Okazaki fragment, where is it? Okay, so thank you, DNA ligase. So I need you all to participate apa yang saya tanya. Alright, so thank you, Razin. Okay, So moving on, if you have question, you can ask. Okay, if you have question, you can ask. If you don't understand, you can also ask lah, kan. So janganlah terima bulat-bulat, kan. So saya maybe goreng awak ke, okay, so you must take note. Okay. Alright, so question number 3, the next question. Name the enzyme that removes primer from the 5' end and replaces them with DNA nucleotides. So the answer would be DNA polymerase 1. So ini senang je, kan. So in the synthesis of leading strand or lagging strand, the primer are made up of RNA nucleotides. Okay, because this process involves DNA replication, you do not want RNA nucleotide in your DNA. So the RNA nucleotides has to be removed because the structure is different. The nucleotides of RNA, it consists what? Pentose sugar bonded to a 5-carbon sugar, which is ribose. Okay, ribose, and then bonded to a base. Okay. So the base can be adenine, guanine, cytosine, and uracil. You do not want uracil in your DNA. You do not want ribose in your DNA. The 5-carbon sugar in DNA should be what? Instead of ribose, it should be what? Dia, apa dia? Instead of ribose, what is the 5-carbon sugar that should be present in the nucleotide of DNA? Dia, Alia. Nitrogenous base, saya tanya pasal ni. Ribose, 5-carbon sugar. 5-carbon sugar dalam RNA adalah ribose. Okay. Kalau dalam nucleotide, DNA, you have the phosphate bonded to a pentose sugar bonded to a base. For DNA, the base will be adenine, guanine, cytosine, and thymine. Okay. So tadi saya cakap ribose dalam RNA ribose ada dalam RNA. Tapi the 5-carbon sugar in DNA, it is not ribose. It is what? Deoxyribose. Which is the long name for DNA apa? Deoxyribonucleic acid, kan? Deoxyribonucleic acid. Kenapa this nucleic acid, we call it as acid? Apa yang menyebabkan molekul ni berasid? It is due to the presence of the phosphate group. Okay, phosphate group. Phosphate group causes the molecule to be acidic due to when it is in aqueous solution, it is in your cell. So it will be ionized. Okay, and it will be ionized, and the molecule, DNA molecule, they are negatively charged. Okay, due to the release of hydrogen ion from the phosphate group. Okay, so nanti saya show you all dah belajar kan chapter 2 on gel electrophoresis. Dah kan? So sifat DNA ni dia bersifat negatif. So that's why when you are running for gel electrophoresis, you have to connect your tank, kan? Tank tu yang mana start dia, this is your DNA, kan? DNA sample, your gel. So you have to connect your tank to the negative end yang dekat dengan DNA tu. On the opposite side, you have to connect to the positive pole. Okay? So sebab apa? Negatif akan bergerak ke arah positif. So DNA molecule, they are negatively charged. Okay, due to the phosphate group. Alright. So okay, so back to the question. So ini saya nak nak bagi asas kefahaman awak pada struktur DNA ni kan. Struktur DNA, the monomer are nucleotides. Okay, so the 5 carbon sugar are deoxyribose. For RNA, the 5 carbon sugar are ribose. So the bases are also different. The RNA, you have uracil. The DNA, you have the thymine. Okay, so that's why the RNA primer, the RNA primer, where it is made up of RNA nucleotide, have to be removed and then replaced with DNA nucleotide. So this is done by the enzyme DNA polymerase 1. If in the question you can't remember or whether it is DNA polymerase 1 or DNA polymerase 3, kan kita ada dua kan yang kita belajar, DNA polymerase 1 and 3. You play safe, you don't write the number. Okay, you just write DNA polymerase, then your answer will be wrong. Okay, so you have to know specifically which enzyme it involves. Okay, so for this one, it is DNA polymerase 1. DNA polymerase 3, the function of DNA polymerase 3 is to add, okay, to add new nucleotides. Okay, add new nucleotides to the primer or to the elongating DNA strand. Okay, so that's why we call it as DNA polymerase 3, because the enzyme will add the new nucleotide at the 3' end of the elongating strand. Okay, the addition of new nucleotide is always at the 3' end. So an easy way of memorizing or understanding the function of these two enzymes is that for DNA polymerase 3, it adds new nucleotide to the 3, kan, dua-dua tiga kan, to the 3' end of the elongating strand. Okay, DNA polymerase 1, remove primer, replace with DNA nucleotide. So you have to know all the enzymes that are required during during DNA replication. Okay, you have to know all the enzymes starting from helicase, and then you have topoisomerase, and then you have single strand binding protein, and then you have primase, you have DNA ligase, DNA polymerase 1, polymerase 3. You have to know all these enzymes. You have to know the function of each one. Okay, so kalau lupa, please go back and do your revision. It is better for you to, after you have done your revision, kan, you do your own notes so that you can understand better, and maybe note awak tu membolehkan awak lebih faham dan ingat, kan. So you are using different colors, for example, so easier for you to memorize. Tapi bio, jangan hafal tanpa faham. So kalau awak hafal tanpa faham, itu yang bila dalam exam dia jadi awak goreng kat situ. Okay, so fahamkan dulu, and then baru awak try untuk ingat slowly apa proses yang terlibat. Okay, alright. So for the next question C, figure 14 shows the final step of translation in protein synthesis. Okay, so translation, where does the process occurs? Jazrin. Tutorial saya bukan sesimpel salin soalan ya. You have to know. Bukan salin soalan, salin jawapan. Translation, you have to know the location where the process occurs. So translation berlaku dekat mana? Ha? Translation diperlukan ribosom. Okay, ribosom tu kilang organel yang terlibat dalam protein synthesis. Tapi exact location in your cell, you have two distinct region, in the nucleus, in the cytoplasm. So translation occurs in which region? Cytoplasm. Okay, so thank you. Okay, so cytoplasm is the location where translation occurs. Same goes, it applies for both, okay, prokaryotic cell and also eukaryotic cell. Before translation is what process? Transcription. Okay, for prokaryotic cell, transcription occurs in which region? Cytoplasm also. So, kena tahulah. Thank you. Okay. So, the process of protein synthesis begins with the synthesis of mRNA first. So, the synthesis of mRNA involves the process of transcription. Okay. So, copying information from DNA in the if it involves eukaryotic cell, it is in the nucleus. But for prokaryotic cell, because prokaryotic cell does not have any nucleus, so the process of both transcription and translation for prokaryotic cell, it occurs in cytoplasm. Eukaryotic cell, it occurs in the transcription occurs in the nucleus, translation occurs in the cytoplasm. Okay, so so that is the basic understanding of transcription and translation. Okay, so after mRNA has been produced, the mRNA, if in eukaryotic cell, it will travel into the cytoplasm. But before the mRNA travels into the cytoplasm, what is the stage that the mRNA has to go through before going out of the nucleus after transcription? Nucleus, your DNA. Okay, through transcription, it produces mRNA, which copies information from a gene in your DNA. Okay, gene 1, for example, produces mRNA. So the molecule is much more smaller, but it will, before going through the cytoplasm, okay, to undergo the process of translation, what is the name of the step that the mRNA has to go through before translation? The name of the process. Fakri Ikwan. Apa proses dia sebelum translation dalam eukaryotic cell? Modifying RNA. Apa nama proses dia sebetulnya? RNA? RNA apa? Okay, RNA processing. Thank you. Okay, so RNA processing. Okay, so RNA processing, it has two stages. Okay, it has two stages, which is what? The removal of apa? The removal of intron and joining the exon, ataupun we call it as RNA splicing. The other one is modifying the two ends of the mRNA by adding what? 5' cap and 3' poly A tail. Okay, so yang you have RNA splicing. Okay, splicing that removes the non-coding region and joining the coding region to form a continuous coding region on the mRNA. Sebab apa? Bila dia nak melibatkan proses translation nanti, the mRNA produced, it must consist only of continuous coding region. Okay, Continuous coding region, apa istilah dia kita panggil dia? Coding region, apa istilah dia? Apa dia? Kita ada istilah dia. Exon. Okay. So, good attempt and thank you. Okay, so exon. Okay, exon. So you have to remove intron. Okay, intron is the non-coding region you have to remove, and then you have to join all the coding region. Okay, all the coding region, so that the mRNA consists only of coding region. Okay. So that is RNA processing. RNA processing only occurs in eukaryotic cell. It does not occur in prokaryotic cell. So maksudnya untuk prokaryotic cell, bila transcription dah hasilkan mRNA, boleh terus proceed kepada translation. Tapi kalau eukaryotic cell, transcription, lepas tu RNA processing, baru translation. So you have to remember that both transcription and RNA processing, in eukaryotic cell, it occurs in the nucleus. Translation sahaja yang berlaku dalam cytoplasm. So itu tadi RNA splicing kan. So the addition of 5' cap and also poly-A tail, it has its own significance. Its function. Okay, which is to protect the mRNA. Okay, so to protect the mRNA in the cytoplasm from being degraded by hydrolytic enzyme in the cytoplasm. That is one of the function of adding the two ends of the mRNA. The other function of adding the two end is that to assist the mRNA binding to the ribosome. Okay, during translation nanti. Okay, one is to protect the mRNA from hydrolytic enzyme, and then second is to assist the attachment of the mRNA to the ribosome, and then it has three functions if I'm not mistaken. The other one is to facilitate, okay, the mRNA movement, okay, to the cytoplasm. So you have to know the significance of RNA processing. Okay, so back to the question. Question number 1, elongation continues until a stop codon in the mRNA reaches the A site. Okay, what is the function of stop codon? Okay, during translation, okay, the codon that will be read is the codon that is in the A site. The ribosome, it has two regions. It has the large ribosomal subunit. It has the small ribosomal subunit. The large ribosomal subunit, it has three sites: the A site, the P site, and also the E site. Okay, amino acid tRNA site, peptidyl tRNA site, exit site. Okay, so you have to know the function of each of these site. Okay, so what is the function of stop codon? Stop codon, when it reaches the A site, it indicates, okay, the termination of translation. It stops the process of translation, or stop the process of polypeptide synthesis. Only 1 mark. Okay, so straightforward questions. Okay, so describe the event that occurs after X binds directly to stop codon at the A site. So when the A site reads the stop codon, there will be a protein, we call it as release factor. So X is release factor. Okay, so this question, it is worth 2 marks. Okay, so you have to explain. When the question asks you to describe, you have to explain how the process occurs. Okay, so the first point, you may include in your answer, X is release factor. Okay, 1 mark. Which causes hydrolysis. Hydrolysis of what? Which is the breaking of bonds. Okay, the breaking of bonds between the last amino acid of the polypeptide and the tRNA. Okay, so that is the function of release factor. Okay, to break bond between the last amino acid and the tRNA at the P site. Okay, so when you break the bond, so now you have the completed polypeptide chain that is ready to be released. Okay, dia dah complete lah, the process of translation has now completed. So after that, the whole assembly that involved in translation will dissociate. Okay, the two ribosomal subunit and also the component of translation assembly dissociate. Okay, so soalan ni 2 markah je. So if you just in your answer you mention release factor breaks the bond between the tRNA and the last amino acid of the polypeptide chain, then you get straight away 2 marks. Okay, so but this type of question, it can worth 3 marks. Okay, so you have to explain a bit. Okay, where you mentioned that the process involved is hydrolysis. Okay, so this is just additional point that you can take note. Okay, if the question 3 marks or 4 marks later. Okay. Okay, so if you have question, you can ask. Okay, jangan terima je. Boleh tanya. Okay. Okay, so the next question, the figure 15 shows a lactose operon. Okay, lactose operon. So the lactose operon is a group of genes, okay, that are found in bacterial cell, and this operon is regulated in the presence of a molecule. Okay, in the presence of molecule, because we are focusing on lactose operon, so it means that this operon, a group of genes ni will only be activated in the presence of lactose. Okay, it means that the operon will be switched on. So you have to know the component of the operon. So the operon will consist of a promoter. So promoter is the binding site for RNA polymerase. And then after that you have the operator. The operator is the binding site for repressor protein. Okay. And then you have the 3 structural genes. Okay. So you have to know the sequence of these genes, which is you have the LAC-Z gene, you have the LAC-Y gene, and also the LAC-A genes. Okay. So this gene, when it codes, okay, for enzymes, okay, for enzymes that involve in the metabolism of lactose. So maksudnya enzyme tu hanya akan dihasilkan dengan adanya lactose dalam sel. Okay, so kita bila awak tengok lac operon ni, dia melibatkan gene yang ada pada bakteria. So in this case is E. coli. There were scientists back then that studies this operon dengan cara macam mana dia grow, the scientists grow the E. coli in a medium. Okay, in a medium, this is E. coli, so dia ada dia punya nucleoid region. Alright, so this is, you can imagine this is, for example, a petri dish that has the medium that contains lactose. Itu je sumber makanan bakteria ni. Okay, itu je sumber makanan bakteria ni. Tak ada sumber lain dah. So bacteria will need to take up the lactose into the cell. Okay, into the cell. So in order to take up lactose into the cell, it requires the enzyme permease. Okay, permease. The enzyme permease is coded by which gene? Hana Sophia, which gene? Permease. Okay, like Y. Okay, so thank you. So kena ingat ni, dia punya enzim yang dihasilkan ataupun dikodkan oleh each gene ni. So permease is the enzyme coded by the LAC-Y gene, which causes lactose to be able to enter into the cell. So basically the function of permease to facilitate the uptake of lactose into the bacterial cell. So once lactose is inside the bacterial cell, Okay, some of the lactose will be converted into its isomer, which is allolactose. Okay, lactose, some of it will be converted into allolactose. Okay, allolactose, dia function sebagai inducer. Okay, inducer. So, induce what? Induce the operon to be switched on. Switched on. Dengan cara apa? Allolactose will bind to the repressor protein. When the repressor protein binds with the allolactose, it changes its shape. So asalnya bila repressor protein ni dalam keadaan shape asal dia akan bind kepada operator. Bila dia bind kepada operator, dia block RNA polymerase from binding to the promoter. So transcription tak boleh nak berlaku. Okay, so tapi dalam keadaan yang bila ada lactose dalam sel, so this inducer molecule or allolactose, okay, bind to the repressor protein, causes the repressor protein to change shape, so therefore it cannot bind to the operator. So when the repressor protein cannot bind to the operator, so the operon is switched on, so the RNA polymerase can bind to the promoter and dia boleh lalu lah dengan lancarnya dengan apa kesan dia? It causes transcription to occur and causes a single mRNA to be synthesized from the 3 structural gene, and the mRNA will undergo the next step, which is translation, to produce the 3 enzyme. So like Z gene produces the enzyme beta galactosidase. Okay, galactosidase. So the lactose that has entered into the bacterial cell needs to be metabolized. Okay, so lactose, its monomer is what? Glucose and galactose. So that is the function of beta galactosidase, which is to break lactose into its monomer glucose and galactose so that the cell can use for cellular respiration to generate energy. Okay, and then the last structural gene is like A, which is to produce the enzyme transacetylase, okay, which function to detoxify other molecules that enters into the bacterial cell. Okay, so okay, so that is the basic understanding on like operon. Okay, so kena tahu bila masa dia akan switch on, bila masa dia akan switch off. Switch off, okay, dengan ketidakhadiran lactose dalam sel. So bila lactose tak ada dalam sel, buat apa sel bakteria tu nak menghasilkan enzim? Tak perlu, kan? Tak perlu. So bila tak perlu hasilkan, so this is to conserve or reserve energy for the bacteria and prevent wastage of energy kat situ lah, kan? So kat situ dengan tak ada je lactose, so repressor protein dalam keadaan shape asal dia boleh bind kepada operator dan block RNA polymerase, and there will be no transcription. Itu bila off. Tak ada lactose dalam sel. Okay, so kalau awak notice kat sini, kat awal ni kita ada satu gene, kita namakan dia sebagai regulatory gene. Okay, regulatory gene. Regulatory gene, you have to take note that it is not part of the operon. Dia bukan sebahagian daripada operon, tapi dia akan mengawal, dialah yang akan mengawal like operon ni. Dengan cara apa? Regulatory gene will produces mRNA that synthesizes the repressor protein. Okay, regulatory gene is not part of the operon. Cuma fungsi dia adalah untuk menghasilkan repressor protein tu. Okay, so identify X tadilah ni kan, sama ada allolactose ataupun isomer of lactose or function as inducer. Okay. Okay, so next, what happened to the repressor protein after X bind? Okay, X ni apa? Allolactose tadilah kan. So allolactose bind to the repressor protein, 2 marks, changes shape. Okay, when it changes shape, it is not active. Okay, it is not active. So when it is not active, it cannot bind to the operator. Okay, so make sure you understand how like operon function. Like operon ni di bawah tajuk apa? Gene regulation kan? Gene regulation. Okay, gene regulation. So di bawah subtopic gene regulation. So the purpose of us learning this like operon untuk kita fahamkan, okay, untuk gene ni diaktifkan, mesti ada satu keadaan yang akan aktifkan gene tersebut. Okay, sama juga dalam sel badan kita ni kan. Semua sel dalam badan kita ni, okay, daripada hujung kaki sampai hujung rambut, semua sel-sel dalam badan awak ni mengandungi genetik material yang sama. Okay, gene yang sama. So, macam mana sesuatu organ tu berfungsi sebagai organ tersebut? Macam contohnya, awak punya pancreas. Okay, pancreas. Pancreas, what is the function of pancreas? To produce digestive enzyme and also hormones that regulate blood glucose level kan. They produce insulin, for example. Okay, insulin dalam pancreas. Tapi sel lain dalam badan awak juga mengandungi gene untuk menghasilkan insulin. Okay, gene dekat pipi awak, gene dekat mata awak, gene dekat tangan, kaki, semua awak akan ada gene yang code for the synthesis of insulin. Tapi kenapa insulin tu tak dihasilkan oleh mata awak contohnya? Sebab dia perlukan certain condition, the cell perlukan certain condition for the gene to be activated. Faham ke? So that's why kita belajar pasal like operon ni untuk fahamkan gene, how gene are regulated, how gene are expressed. Boleh Can or cannot? Okay. Okay, so the next question, state the function of enzyme Q. So ini I dah bagi dah tadi kan. So Q, enzyme permease, which is to facilitate the uptake of lactose into cell. Kalau dalam soalan awak jawab, okay, awak jawab facilitate the uptake of lactose. Sampai sini je awak jawab. Jawapan awak boleh jadi salah. Okay, facilitate the uptake of lactose by what? Into where? Kan, into the bacterial cell. Okay. So that's why pada awal tadi kan saya tekankan you all kena faham apa yang you all baca. Kan. So kalau awak just hafal, maybe awak hafal separuh jalan kat situ kan. Okay, permease uptake of lactose. Lactose uptake kat mana, untuk apa kan? So facilitate the uptake of lactose into bacterial cell so that bacterial cell can use as source of energy, can undergo for metabolism kan. Alright. Okay, so the next question 5. Okay, this one we can go through quickly. So figure 16 represents a tRNA molecule. Okay, so tRNA awak tengok dia macam bila kita belajar RNA, selama ni awak belajar dia single strand kan, single stranded. Okay, DNA je yang double stranded. Tapi awak nampak kat sini, ada macam double strand kat sini. Sebab apa? From the single strand, it folds up, okay, into a clover leaf shape. Kan? So, this sebenarnya single strand. Okay, the RNA is actually single strand molecule, but it folds into specific shape. So, for the tRNA, it has two major region, which is the X region is the amino acid binding site. Okay, so every tRNA will carry specific amino acid. We have 20 different amino acid. Okay, 20 different amino acid, and this amino acid will be carried by different tRNA based on the anticodon on the tRNA. Okay, so the anticodon will be complement. It is complement to the mRNA codon. Okay, so during translation, when when the ribosome reads the mRNA kan at the A site, so A site ada codon apa, so nanti akan datang tRNA bawa specific amino acid that has anticodon that is complement to the codon at the A site. Okay, so name the molecule that is attached to X during protein synthesis. It is amino acid. Jadi saya punya jawapan ni as short form ya. Bila dalam exam awak jangan tulis AA. You have to write it full amino acid. Okay, so second, name the type of bond label Y. Okay, so tRNA. Okay, bila dia dalam keadaan adjacent macam tu, they are bases kat sini kan. Maybe kat sini ada adenine, maybe kat sini ada uracil. So the bases between adenine and uracil will be attached together through the formation of hydrogen bond. Kat sini maybe ada guanine, sini maybe ada cytosine. So the bases that are adjacent to each other at this region are held by hydrogen bond. Okay. Kalau tengok kat sini kan, dalam soalan ni saya bagi tips sikit. Dia cakap apa? Name the type of bond label Y. Dalam soalan dia dah mention perkataan bond. Okay. So kalau awak just tulis hydrogen pun dah cukup. Okay. Ataupun dia tengoklah kan, kalau awak tulis H, tengok kadar kesimpatian pemeriksa tu. Dia boleh bagi betul juga. Okay. Okay, so what is anticodon? So this one is so if you notice kan, soalan nombor 5 ni soalan yang awal-awal. Okay, soalan awal-awal. So bila soalan awal-awal, bukan anak-anak yang seterusnya, dia bagi soalan yang straightforward je. Okay lah, level yang senang. But if anak-anak yang seterusnya, dia akan menjadi semakin susah macam tu. Okay. So kita soalan dia ada 3 level lah. Soalan straight from the nota. Soalan yang awak kena fikir sikit, dia bagi soalan yang level yang ketiga, dia bagi satu situation awak kena applykan knowledge awak, apa yang awak dah belajar. Okay, so 3 level. Okay, 3 level. Kalau awak perasan awal-awal semester kan, awak punya lecturer ada bagi dalam slide taklimat, dia cakap level, if you notice lah, tapi tak apalah. Okay, so what is anticodon? So anticodon, you just give the definition, which is a triplet basis on tRNA that is complementary to the mRNA codon. So do you have any question? So ini jap the next question for you to differentiate. So bila soalan differentiate, you have to put your answer in a table. Tapi luckily for this question, dia dah bagi siap-siap table. Okay, so just fill up the table. Kalau soalan dia 2 markah untuk difference dan disuruh awak bagi 2 perbezaan, if you give more than 2, jawapan pertama, jawapan kedua, jawapan ketiga, the first answer is wrong, the second answer is right, the third answer is right, examiner akan mark hanya the first 2 sahaja. Okay, so it means that you get only 1 mark right. Sebab apa? Sebab the konsep dia adalah pemeriksa nak you all tahu, pemeriksa nak memastikan you all faham ke tak apa yang you all jawab ni. Bukan main teka je. Okay, tekaan pertama salah, tekaan kedua salah, ketiga dan keempat betul. Okay, so you must precisely know what you are answering. Macam tu. Okay, so ini just a note lah kan. So you can any of this point you can get 2 marks. Okay, satu perbezaan yang betul, 1 markah. Okay, satu perbezaan yang betul, 1 markah. Okay, so dia mesti tally. Jadi kalau describe shape, clover, leaf shape untuk tRNA, tapi tiba-tiba kat sini awak cakap apa? Tak ada amino acid binding site. So you are not comparing apple to apple. If you are not comparing apple to apple, walaupun point dia betul, jawapan awak salah. Macam tu. Itu cara macam mana soalan awak ditanda, kan. So for comparison, soalan yang suruh compare, you must compare benda yang sama, kan, apple to apple, then only you can get 1 mark. Okay, so I guess we can end our session for today. So do you have any questions? Dah nak ke pukul 10 dah. Tak ada soalan? Kalau tak ada soalan, so I guess we can end our session. For next week tutorial, saya dah takkan discuss dah soalan chapter 1 ni. Okay, so I hope you have done the question, the exercise for chapter 1 ni. Sebab tu saya tanya, keep on asking. You all ada soalan tak? Ada soalan tak kan? So mana yang you all tak faham, boleh tanya. Okay, sebab next week, saya nak kita move on to chapter 2 punya soalan. Okay, so make sure before the tutorial session, you have already done the question. Okay? Okay class, so I guess we can end our session. So thank you. Okay. Okay.